Okay, so this is the um, second chapter, and after this, today we're going to look at doing a turn this on. So after this, we're going to look at the lab as well. Didn't work. Let's try that again. Slideshow. Slideshow. From beginning, I'm going to make sure that this stops and start it over again so we get that slideshow going. Okay, there we go. Okay, so we're going to look at chapter 2. You should also look at the pre-lecture video for chapter 2. And then additionally, everybody is responsible for looking at the lab for Thursday. The lab, we have 3 groups of 2 people. I put Ruth in a group of her own because she's going to be not able to come to lab until later. And then John's in a group of his own. But you've all been assigned to look at a genetic mutation that affects a checkpoint protein. And as you do that, there are some questions to answer on the lab. Those are due next week on Tuesday. What you're going to do is design a fact sheet or make a short video. Those fact sheets are probably not going to be more than about a page or 2, or a video that's about 5 minutes or so. There's a few other questions on the lab that you can answer on a separate sheet, and all of those should be turned into the assignment box by next week, Tuesday, and we will talk about them as well in class. So I will then assign you guys to learn those things so that you'll have that information as well for the next test. Okay, so what we're going to talk about today is meiosis, as well as the checkpoints that you find in the cell cycle. So those are the 2 things that we need to talk about today. You should learn mitosis by looking at the slides and the lecture for mitosis. It's in the pre-lecture part of this whole thing. Realize that there is also a meiosis assignment due that you should be able to do and turn in. There is an assignment box for that actually is, I changed it so that it's not due quite yet, so that it will actually be due next week on Tuesday. Okay, to start with, if you look at chromosomes in a karyotype, you can see the distinct parts of the chromosome. So on each of the chromosomes, you have a centromere. That's not to be confused with the centrioles or the centrosome, which are the microtubule organizing centers. The centromeres are on the chromosomes. The kinetochore is a part of a group of proteins that attaches to the centromere that allows the centromere to attach to the kinetochore microtubules. The centromere is not necessarily in the center of the chromosome, but it is an area that has a distinct sequence that basically allows for central kinetochore proteins to attach. This is a chromosome on one side. It's been replicated to have 2 sister chromatids. When you look at chromosomes in a karyotype, they are in their most compacted form, and you can distinctly see the 2 sister chromatids. Okay? At the end of the chromosomes, you should have telomeres at both ends, which are the ends of the chromosome. Karyotypes are made by looking at chromosomes after they divided and when they're in the process of doing mitosis, so they're already compacted. And you can see each of these so each of these sets here is a set of homologous chromosomes. So under one here, you've got a pair of homologous chromosomes, and each of those homologous chromosomes has 2 sister chromatids. Really important thing to understand is homologous chromosomes have the same genes in the same order, but not necessarily the same alleles. Sister chromatids have exactly the same genes and exactly the same alleles. Okay? So if you have a heterozygote, big A, little a, each of the sister chromatids from this chromosome should have the big A's. Each of the sister chromatids from this chromosome should have the little a's. Okay? That's really important to understand. And the meiosis thing that we do today will basically talk about that. A diploid cell basically has two homologous chromosomes. Each chromosome has two alleles for every gene. So diploid cell, basically, you have one set of chromosomes from mom, one set of chromosomes from dad. They have exactly the same genes on them, but they may have different alleles. Okay, a diploid cell is also called a 2N cell. And if your 2N number, let's say your 2N number equals 8, that means that you have 4 pairs of homologous chromosomes. When you go through S phase, each homologous chromosome then has 2 sister chromatids that are connected together. They're connected together at the centromere, but they're also connected together at least loosely all the way along. And that's where that cohesed comes into place that we talked about in the pre-lecture talk. Okay, so then after S phase, you have each pair of homologous chromosomes will have 2 sister chromatids. Okay, during mitosis, each of these homologous chromosomes lines up separately on the metaphase plate. During the first stage of meiosis, the homologous chromosomes line up together next to each other on the metaphase plate. Okay, telomeres, as I said before, are the parts at the ends of the chromosomes. Now, if you look at a normal cell cycle, interphase makes up the vast majority of that cell cycle. Look at this. This one's like 25 hours or 24 hours, and interphase makes up all but 1 hour of that process. Interphase consists of G1 phase, which is when the cells prepare to grow and go through S phase. Then you have S phase when the chromosomes are replicated. Then you have M phase, or G2 phase, in which the cells prepare for mitosis. And then you have a really short division, which basically has that prophase, metaphase, anaphase, telophase, and cytokinesis. And that's mitosis. That's the part that we spend a lot of time talking about, but it's actually the shortest part of the cell cycle. Now, during this whole interphase, and even during mitosis, we have a number of different checkpoints that allows the cells to check and see how they're doing and make sure that they have the proper materials and the proper signals to allow them to grow. So there are a number of different cell cycle checkpoints. The big one is in G1. And during G1, the cells basically ask themselves, are they ready to grow? Do they have the right signals to grow? There are also checkpoints in S phase, which basically makes sure that the DNA has been replicated once and only once. We'll talk about that one when we talk about replication. There's another one in M phase, which is the spindle checkpoint, which basically asks whether or not the chromosomes are properly attached to the spindle. Okay? Now realize that and then there's a fourth one throughout the cell cycle. It's several stages, which asks whether or not the DNA has been damaged. And if the DNA has been damaged, the cell cycle will stop, and you won't go any further. Okay? Now realize that in an adult organism, most of your cells are actually in G0. G0 is replication arrest. The cells are going through metabolism, they're making proteins, but they're not going to divide, they're not going to go through S phase again. The only cells in an adult human's body that are active in terms of mitosis are going to be stem cells. And then you have germ cells that are active for meiosis. Okay? Every tissue in your body will have some stem cells that can replicate. Now, to monitor how the cells go through the cell cycle, and to allow for the cells to stop at checkpoints, you have cyclins and CDKs. CDKs are cyclin-dependent kinases. They are only active when they're attached to a cyclin. the cyclin will direct their activity so that they will find the proper targets. Okay? Cyclins are only produced just before they're needed, and they're rapidly degraded after they are needed. They then allow the CDKs to phosphorylate targets that will move the cell into the next phase of the cell cycle. Once they're into the next phase, the cyclins get rapidly degraded. This is what the progression of cyclins look like. In the G1 phase of the cell cycle, cyclin D is active. Don't worry about the type of CDK, but the cyclin D is active in the G1 phase of the cell cycle. As you get into S phase, you get cyclin E being really active. To get through S phase and into G2 phase, you need cyclin A. Cyclin A is also active in G2 phase. Cyclin B is needed to get you into M phase. And to get you through M phase, you actually need to destroy cyclin B. Cyclin B is going to be destroyed around the time of metaphase, moving into anaphase, and we'll see that in a minute. Okay, so we're going to look at two checkpoints. The first one is the G1 phase checkpoint. And the G1 phase checkpoint is the one that is activated by cyclin D, or that is controlled by cyclin D. Cyclin D, basically its job is to phosphorylate a protein that's called RB. RB is a negative regulator of a transcription factor known as E2F. So normally what happens in the cell cycle in G1 phase is E2F is sitting on its promoter, and RB is sitting on top of E2F, and is preventing E2F from activating genes that would move the cell into S phase. Okay? Now, in G1 phase, the cell should be getting signals to divide from growth factors and from other things. If those signals are in place, cyclin D will phosphorylate, cyclin D and its CDK will phosphorylate phosphorylate RB, and so will cyclin E, the next guy down during that comes up in the cell cycle. It will also phosphorylate RB. When that happens, RB needs to leave E2F. It's inhibited. E2F will stay on the promoter, but now it can activate the genes that will produce proteins that will get us into S phase. Okay? If the cell does not receive growth signals, or if it doesn't have enough nutrients, RB will remain unphosphorylated. Cyclin D will not build up to the level where it can phosphorylate RB. If you don't phosphorylate RB, you don't move out of G1 phase and you don't get into S phase. So the cell will use RB and the level of cyclin D to determine whether it's ready for S phase. And only when it's ready for S phase will cyclin D phosphorylate RB, allowing E2F to transcribe genes whose proteins are involved in S phase. When these proteins are produced, S phase will start. The spindle checkpoint is the checkpoint that basically checks to make sure that all of your chromosomes, all your sister chromatids, are attached to the kinetochore microtubules before you move from metaphase into anaphase. So remember, metaphase, the sister chromatids are lined up on the metaphase plate. When you get to anaphase, the cohesion between those two sister chromatids is destroyed. And what happens then is the sister chromatids move towards the opposite poles and you go into anaphase. Now, if you have a set of sister chromatids that is not connected on both sides to the microtubules, what will happen during anaphase is that both of those sister chromatids will move towards one pole. One cell will end up with extra chromosomes, and the other cell will end up losing a chromosome. Okay, that's that segregation error that you see down here. That's non-disjunction is another word for that. And what happens then is you have a cell that loses genetic material and a cell that gains genetic material. So in order to prevent that from happening, we need to make sure that all of the spindle microtubules all of the kinetochore microtubules are in place and attached to the kinetochores before we break down that cohesin that allows the chromatids to stay together. Okay, so if you look at this thing in the middle here is suffrin, or is cohesin, and cohesin basically is a ring protein that connects together the sister chromatids. It's found more heavily in towards the middle of the chromosome where the centromere is, wherever the centromere is. But it is also found all the way along the edge of the chromosomes, connecting the sister chromatids. When you move from metaphase to anaphase, sufferase acts as a molecular scissors to cleave the cohesin protein so that the two sister chromatids can move to the opposite poles. Okay? So what the cell has is a way of sensing when the kinetochores of microtubules are connected to the kinetochores. And that's going to be sensed by the kinetochore proteins, including this protein called MAD2 over here. And if those proteins are not in place and not connected to the kinetochore microtubules, you will not be able to move from metaphase into anaphase. Okay? Moving from metaphase into anaphase basically requires this complex here, which is called the anaphase promoting complex. And it does a couple of things. First, it basically breaks down cyclin B, which you need to move into anaphase. And the other thing that it does is it basically causes sufferase to cleave cohesin. And once it does that, it allows the cells to move from metaphase into anaphase. If that does not happen, if you don't have good connections between the kinetochores and the microtubules, then the cells are going to stay in metaphase until they basically are signaled to go through apoptosis. So if they can't finish up the cell cycle, they will go through apoptosis. Okay, so that's all I have to talk about in terms of cell cycle checkpoints. The last thing that we're going to talk about is sexual reproduction and meiosis. So realize meiosis basically allows you to go from a 2N cell to a 1N cell. And then the 1N cell then becomes the gametes. The gametes then will recombine to form a fertilized egg. And those fertilized eggs then become a zygote, which then can become an organism. Okay? Now, there are different organisms out there. Some of them basically have gametes that are the same for both females and males. Those are called isogamies or isogamous. Heterogamous is what we are, and that means that our gametes are very different from one another. Our eggs are very big, our sperms are very small. They look very different from one another, and the sperm will fertilize the egg, and that will basically make the zygote that then becomes the adult organism eventually. So meiosis basically has two stages, meiosis 1 and meiosis 2. We start with prophase, and there are multiple different stages in prophase. But the biggest thing in prophase is the chromosomes are going to condense, they're going to come together. Unlike mitosis and meiosis, the two homologous chromosomes have to line up together, and actually they're fused together in this stage. Crossing over can occur between those chromosomes, and the nuclear membrane is going to fragment, and the spindle apparatus is going to form in this stage of prophase. So let's look briefly at those 5 stages of prophase. What happens in the first of those stages, the Lapertina, is that the chromosomes are going to condense, and they're going to start to find each other. So you've got your 2 versions of each chromosomes, your 2 homologous chromosomes lining up next to each other. Then what happens next is the synaptonemal complex is going to form. That forms in the next stage, digotina, and that basically fuses together your homologous chromosomes. In pachytina, going into diplotina, you start to have crossing over occur. I think there's a little bit of confusion sometimes in which stage exactly it occurs, but it definitely starts in Pachytina. And what can happen? This can happen anywhere along the chromosome. You can have one strand from one homologous chromosome exchanging information with the other homologous chromosome. That allows for further genetic heterogeneity as that occurs. What happens in Diploctema basically is the synaptonema complex starts to break down. You can see the crossing over has already occurred here, so you really don't need it as much anymore. And then we end up at the end of prophase 1, but those homologous chromosomes will be next to one another going into the rest of the stages so that we can then separate them off from each other. Here's the synaptonema complex forming between the homologous chromosomes, allowing for, you need to have this happen really accurately, so when you exchange genetic information, you don't cut off a gene in the middle of a gene, that you have a really good matching between these chromosomes, and that way you can assure that you have a faithful connection between them, and that they're not going to lose or gain genetic material if they do crossing over. At the end of this, at the end of prophase, then we go into metaphase. In metaphase, the homologous chromosomes are lined up on the metaphase plate. The kinetochore microtubules connect to one homologous chromosome on each side. Okay? And then when we go into anaphase, the homologous chromosomes move away from each other. As we go through this, then, you end up with two different cells. You go through telophase and cytokinesis, two cells next to one another, and both of those cells are now one end cell. They still have sister chromatids, so they still have basically the same amount of genetic material as any normal cell would have, but they only have one copy of each allele. Okay, and realize that's pretty random. So you may get, so your cells have one copy of every chromosome from mom and one copy of every chromosome from dad, but when you make germ cells, those randomly assort. So you may have chromosome 1 from mom ending up in one of your germ cells, but that may be matched with chromosome 2 from dad. So there's nothing special about those chromosomes from mom or from dad. Your cells don't care which ones they find, as long as they only end up with one chromosome out of each pair of homologous chromosomes. Okay, when you go into meiosis 2, you go through, again, you recondense the DNA. This could happen really quickly after meiosis 1, or it could, this could really happen really quickly after meiosis 1, or it could happen a long time later on. But then we go through meiosis 2, the chromosomes recondense, the spindle reforms, metaphase, the sister chromatids line up on the metaphase plate. Anaphase, you're going to split them off. The sister chromatids are going to move to opposite poles. And then telophase, you basically are going to form two more cells. And these two cells are also one end cells, right? But they're one end cells that now only consist of one of those two sister chromatids for each chromosome, right? So what happens at the end of this in meiosis 2, if you're producing sperm, you have 4 sperm, right? Each of these sperm will have one end chromosome number. For every chromosome that you have, every homologous chromosome, 2 of these will have one of those pair, and the other 2 will have the opposite one of those pair. Okay? And then you form 4 sperm from that. Any of those 4 sperm could fertilize an egg, with a few exceptions, right? Sometimes you get some that are not quite as good as others. When you do division to make an oocyte, you start off with a set of homologous chromosomes. In the first division, that divides asymmetrically. So you end up with one big cell and one little cell that we call the polar body. So your secondary oocyte is a 1N cell with two sister chromatids for every chromosome. The polar body is the same. It has the opposite genetic material from the secondary oocyte. So if the secondary oocyte has chromosome 1 that you got from your mother, the polar body would have the chromosome 1 that you got from your father. Okay? You then go through a second division, and it's really thought that that polar body, that first polar body, may not go through a second division. So it might just be the secondary oocyte that does that. The sister chromatids basically divide into two different cells. You end up with a second polar body, which is exactly the same as the egg. So it's got exactly the same genetic material as the egg. It's an asymmetrical division again. So the egg ends up with more cytoplasm than either of the polar bodies. So basically, human females end up with one egg and two polar bodies. This polar body down here, the first polar body, has two sister chromatids for every chromosome, and it has the opposite chromosomes from what the egg has. The second polar body has one sister chromatid for every chromosome, and it has the same genetic material as you would find in the egg. Why is that important? Because you could actually look at these polar bodies to determine what the egg looks like. So if you're trying to do a genetic test to determine what that egg has, in order to determine whether the child has inherited a genetic disease, you want to know which polar body you're looking at. Okay? The egg cell is haploid, the sperm cell is haploid. When they come together, they form a new diploid organism. Okay? Non-disjunction occurs when the homologous chromosome fails to separate properly in meiosis 1, or when you have a inability of the sister chromatids to separate properly in meiosis 2. That will result in more or less than the expected number of chromosomes in the resulting cells. So here's non-disjunction in meiosis 1. What happens in that case, the two homologous chromosomes go off in one direction. This cell over here is lacking any of those homologous chromosomes. At the end, you'll end up with two of the gametes that have an extra chromosome and two of the gametes that are lacking that chromosome altogether. Okay? If it happens in meiosis 2, you end up with two normal cells and two cells, one of which gain a chromosome and one of which loses a chromosome. Now, in any case, if one of these becomes the egg cell or the sperm cell that produces a new organism, that organism may be lacking a chromosome or may have an extra chromosome. Most of the time, that's fatal. And in fact, that's one of the most common ways that you could have a miscarriage, is to have a child with a chromosomal defect. So fetuses with chromosomal defects very often will die before birth. They're not necessarily viable. One exception to that is trisomy 21, which you have 3 copies of the 21st chromosome, and that forms Down syndrome, which basically is a somewhat severe problem, but it does produce viable children that then can grow into adults. So trisomy 21 is one exception to that. You also can have an exception when you can gain or lose X's or Y chromosomes, and we'll talk about that in chapter 4. Okay, sex chromosomes, obviously. So we have 22 pairs of autosomes and 2 pairs of two sex chromosomes, 1 pair of sex chromosomes. And obviously, you know that XX equals females, XY equals males, with some exceptions that we'll talk about in chapter 4. Okay? The last thing to talk about quickly is the chromosomal theory of inheritance, which basically says that because genes are on chromosomes, if you have 2 genes that are on the same chromosome, you will tend to inherit those alleles together unless there's crossing over between those genes. So for example, if the gene for blue color, or if the gene for eye color is closely linked on a chromosome to the gene for blood type, you will either inherit, let's say your father's blue eyes and his type B blood type, or your mother's brown eye allele and her type A blood type. Those things will be together. And we'll talk about that in chapter 7 when we talk about genetic linkages. Okay? So that's basically it for this chapter. I'm going to stop here and I'm going to put this up, and then we'll move on to Mendelian genetics next time. Okay, so let me stop the share and let me end this meeting.